there.
Yeah, push that.
Okay.
There you go.
Well,
we can see your slides.
okay, thank you. Well, I would like to thank you, Serena and Georgia, and all people who've allowed me to present some of the data in that species that is really dear to us as humans, and is not very well known. Lisette has mentioned electrophysiological recordings just for usually neurological conditions as epilepsy of the temporal lobe, but there are other conditions in which this region can be also of interest. So I would like first to present Rafael Lorente de No. This picture was taken by Larry Swanson while he was working at the Department of ORL in St. Louis University. And yeah, I think it's quite interesting and quite fascinating that this person was not particularly interested in the hippocampus. He was interested in the auditory system, where he produced very interesting physiological studies and so on. So we move just specifically to the region of interest, and we can see a section stained with Nisl of the human hippocampus and showing the different strata at the CA2 level, which is exactly similar to the other CA1 fields of the hippocampus. So we start by the alveus, which is just turning into the pial surface. That will be the surface that borders the cavity of the lateral ventricle, the temporal horn of the lateral ventricle, and then there is just fibers, so the density of neurons is very small. Then we have the stratum oriens, which has also low density or lower density of neurons. Then we enter into the principal cell layer, the pyramidal cell layer. And then we have another cell sparse stratum, which is the stratum radiatum, and then to end with the lacunosum molecularum. So in that respect, it's exactly the same as any other hippocampal field. And this structure, this layering, is conserved along the rostrocaudal axis of the hippocampus. So we can see here something that we produced many years ago. Is in the human nervous system, the book by Paxinos and Jürgen May, and we can see the different parts of the hippocampus with different fields along from the beginning of the hippocampal head. We have here the uncus, the gyrus uncinatus is called, and then we can see the end of the uncus right here, and then we continue with the body of the hippocampus, and then we reach the tail of the hippocampus in these two last sections. So here is indicated by the dark segments the extent of CA2. Notice that the very rostral part of the hippocampus, there is no CA2. And one thing that I have found in the literature is that people place CA2 in front of CA3. And it's in our observations, many cases, more than 100 human cases, it's just the opposite. It's the other way around. It's all continuous CA3, and further down or further caudal is where CA2 starts. And then it's continue, and it's exactly symmetrical when the dentate gyrus opens like in two facing here, joined by CA3. And then this specular image is condensed, but just this is at this level is only CA3, and the gyrus intralimbicus, that is the end of the uncus, is only CA3. But CA2 continues into the body. That will be the anterior part of the body, and it continues all the way down, and then it's elongated because of the obliquity of the section. It was mentioned a couple of times before that the plane of section is quite important in order to understand the extension of CA2. And we have just the size of the hippocampus, from the beginning, from the frontotemporal junction, but there will be the anterior limit of the subiculum, where we start just considering the beginning of the hippocampus, and to the end. That's exactly 4 centimeters. Well, and it keeps much of this size, at least in control cases. In Alzheimer's or other neurodegenerative diseases is not the same. It decreases. So I'm going to show a series of sections from which we're taking the anterior schemes and approximately the level that corresponds to each one. So this is the hippocampal head, that you can see that CA1 is the field that predominates. A little bit of CA3 we can be there. It's not pointed in the picture, the extent of each one of the fields. And then we have the transition to the subiculum and to the entorhinal cortex. We move now towards the end of the uncus. It's not quite, but it's, you can see that now we can see CA2, and it can be appreciated on both sides. So it's fairly consistent in that way, and that we reach the caudal part of the entorhinal cortex. And moving slightly backwards, we notice here the presence of the lateral geniculate nucleus, which is a good indicator of the extent of the body of the hippocampus. That's a useful landmark, and in particular for neuroimaging studies. So we continue just indicating the extent of CA2. I mean, in this picture cannot be appreciated the details, but just the location of CA2. And then we have also CA2 with CA3 at the very end. This is the tail of the hippocampus. You can see how convoluted is the granular cell layer of the dentate gyrus, although it can be seen and recognized every part of them. So this belongs to one communication in the hippocampus. This is a special issue, which is now in press. And we show exactly the same, the series, the beginning of the hippocampus, entering into the head of the hippocampus. That's the beginning of the dentate gyrus. And then the CA3, and then the other fields of the hippocampus. And what I was mentioning before, that CA3 is continuous, and then a little further caudally is where CA2 starts. And then we have the end of the uncus right here. So we continue more or less the same length and the same dimensions. Continue backwards in this part. There will be the caudal part of the body, beginning of the tail right here. And then you can see how an indication of the tail is that the dentate gyrus loses this perfectly rounded and with like 2 arms cell layer, and then becomes irregular, you can see here, and in particular at the end with the tail of the hippocampus. In every place, it's present CA2. So it's a field that goes all the way. How to separate? Because we use basically the Nissl stain. Then we have here the low power of this part of the CA2 and the remaining of the hippocampus, and we place on one side that would be the limit with CA1, and that will be the limit with CA3. So you can see the number of neurons of CA2 is much larger than any other species that has been shown in the previous presentations, even when it's relatively denser at the outer part of the principal cell layer, the pyramidal cell layer. Nonetheless, towards the deep part, it's also populated, although in a lesser density, obviously, but it makes a row of 6 of 8 cells very, very easy. And then how to separate what is and what is not the CA2. This is also an old photograph. This is a team stain. You can see how the mossy fibers is stained by the presence of zinc. It's a very useful indication of the extent of the mossy fibers, and then where it stops, and then what we consider is CA2. Then we have another marker, is neurofilament, non-phosphorylated neurofilament, is SMI32 in the monkey. And then we have also the delimitation of this pyramidal cell layer that stops at CA2, and then it comes again at the cerviculum level. This is acetylcholine, I'm sorry. Yes, this is acetylcholinesterase, is a marker that goes beyond the limit of the mossy fiber and then extends on CA2. And you notice how nicely it delimitates an oblique border. That's another constant feature. Just CA2 with CA3 is an oblique border. CA2 with CA1 is again an oblique border. And the neurons overlap. So the difference between CA2 and CA1, a very useful one, is the size of the neurons. And between CA2 and CA3, the size of the neurons is not any longer useful, but just the organization, the columnar organization of CA3 is much nicer than not in CA2. Obviously, this is a very subjective criterion, and it may change from one observer to another. And this is the SMI32 in human. You can see the difference. CA2 and CA1 is not stained in the monkey, but it's continuous in the human. So those are differences in the expression. Obviously, what has been worked in the gene expression ultimately, but we don't know almost anything on the expression in the human CA2. This comes from another study, which this is the correlation between very high resolution MRI, 9.4 Teslas, and the reconstruction in the coronal, in the horizontal, and then the volume rendering of the shape of the hippocampus. So you notice in green that it's the extent is more or less constant. Look, it thins a little bit more caudally, but you can see that extends from the rostral to caudal end, and although with small irregularities that obviously they come from the judgment of the extent of CA2 in the coronal plane is very consistent. And now I would like to show you how things are at different rostrocaudal levels that will be equivalent to the septotemporal level. And we have to keep in mind very clearly that the temporal level in the rodent is the equivalent to the hippocampal head. And then the extent, the middle extent of the hippocampus, the body of the hippocampus, will be the equivalent to the body of the hippocampus in humans. And then the septal pole will be the caudal part of the hippocampus. So it's interesting to know the rostrocaudal distribution. And we can see here, this is a case of frontotemporal dementia, very serious case with a lot of impact in the cognitive abilities of this person. And this is rostrally at the level of the head of the hippocampus. You can see that there is positivity. And then we move to the beginning of the body, and you can see how it increases. And it shows all this material, this neurofibrillary material deposited along with the deposit of the neurofibrillary tangles inside the neurons. And then we move towards the caudal part of the body, and then at the tail of the hippocampus. So the tail, as I have said, is the equivalent to the septal part of the rodent and other species hippocampus. So there is a difference in the density of damage, or the density of neuropathological tau material, tau protein material is this stain. And obviously that calls for different susceptibility among different neuropathological conditions. And then I would like to show a little bit of the monkey studies where we localized the plane of in the monkey brain with MRI, and then we have the line from the anterior to the posterior commissure, and then what we used as reference to enter into the entorhinal cortex to place tracers. That's something that is still under study. This is an example of the deposit. This is lectin, BBA, and we can see how it covers part of the entorhinal cortex that will go from here up to here. This is the rhinal sulcus. So all that region is the entorhinal cortex, and then it passes and diffuses, obviously, a little bit on the overlying amygdala. That's the amygdaloid complex. And we can see the nasal stain, so you can judge by yourself the extent of the injection. This is another example at a slightly more caudal level. We have here the beginning of the hippocampus, even a little bit of the beginning of the dentate gyrus, just to, I mean, the monkey hippocampal head is much simpler than not in the human, because it lacks these digitations of the hippocampus that make that complex shape of the head into the coronal sections. And we have an example of the tail of the hippocampus. So, I'm sorry, the caudal part of the entorhinal cortex and the end of the uncus. So you can see the projection. That's what we were looking for. The projection to the stratum lacunosum moleculae, the molecular layer of the dentate gyrus is perfectly marked. And then we have here the projection from layer 2 neurons of the entorhinal cortex to the stratum lacunosum moleculae. But significantly, we have here and this other part which is more extensive, and that extends in the stratum radiatum specifically, and is not seen on more proximal parts to the dentate as CA3 or near more distal parts on CA1. That's what we are examining now with the list set in this study, which is under analysis. And then the conclusion was that the rostral part of the entorhinal cortex projects little to this part. So we can see the outline of the end of the domestic fibers, that will be the stratum radiatum. At the intermediate, it increases significantly, and then at the tail is also less prominent than not in other topographical regions of the entorhinal cortex. Those are the equivalent, the adjacent nasal levels, so you can look at perfectly the extent of CA2 and then the other fields. And that's what I wanted to share with you, an overview of the human hippocampus, how little we know, but how important it is in pathology, and considering other conditions that may affect the presumed functions in humans. So, of course, we know that in the rodent, the social behavior, the social memory, aggression, as it has been said, but we don't know how it works in humans. And considering the variability among different animals, I guess that we can find surprises when it is studied more in detail. And thank you again for the invitation, and hope that any questions will be very nice to answer if I can. Thank you.